or a Tuesday, but I don't think that's reasonable because I'd love to get over the high spots in this chapter that we're working on. So we're going to probably put that off till Wednesday of next week for the in-class component.
Thursday.
The Thursday. And we're going to bounce through some of the high spots on this plant growth and development chapter today. I had started on it a little bit the last time we got together and got tangled up and didn't get along as well as I'd intended to. So, but we're going to talk about it today with the plant growth and development and the effect that light has on plants. And some plants compensate for reduced light intensity or increased reflection by what is called a heliotropic movement. And that is defined as the movement that plants have on their own to make them either more receptive to light or more resistant to light. If the light intensity happens to be too great for this particular plant that we might be speaking of, the leaves may turn at an angle where the intensity is not as great. So as we talk about this, a good example of a heliotropic movement is a plant that used to be done primarily as an ornamental, but it has become a significant commercial crop. And some of you have probably witnessed an acreage of sunflowers this past summer. I know there was a farm just up north of where we are at Patrick Henry that had about a 35-acre field of sunflowers. But the sunflower itself will exhibit what we speak of as heliotropic movement, in that the head or the flower component constantly turns to maximize the amount of sunlight that affects that flower component. And that's why it gets its name, the sunflower, because of the heliotropic movements that exist. And a lot of that is attributed to light quality. The sunflower wants a maximum amount of light, so it does that. Now we spoke a minute Tuesday about the photosynthetically active radiation, the light rays that are between 440 and 700 nanometers. And nanometer is the unit that we use to measure the light intensity. But the blue light in the spectrum is at the 440 nanometer range, and then red light is on up towards 700, 650 to 700. When we did this lettuce project here on campus from the grant that we got from USDA, we actually constructed some LED panels using red and blue LEDs. We couldn't find commercial lights that were acceptable for us to use for that project. So we actually took a piece of pegboard, something as simple as a pegboard, and put a 2x2 frame around it, and we would insert red light, then a blue light, and a red light, and we were able to tie those in together, and we used a power source to actually, we made a 4x8 foot panel to use as a part of this light or this experiment that we're doing using growing lettuce in some of these old buildings around here. So, but as we talk about light quality, the blue and red light are the primary lights in the spectrum of our natural light that drive our photosynthetic process. The green light on a green leaf is not going to, just common sense, it doesn't do any good. It's not effective. It's usually reflected. So that's just what we're talking about with light quality. Okay? All right.
Yeah.
Good deal.
I nearly died last night, but
We'll talk about that later. All right. As we talk about light quality, the term photomorphogenesis comes up. There are a lot of words that we deal with in this introductory plant class, and photomorphogenesis is what it says. It's a response of light to make differences in the plant shape and form. So photomorphogenic responses are changes to the shape of a plant. And that's greatly affected by light intensity. If you have light that is of a poor quality or low intensity, generally your plants that are growing in that situation tend to be weakened. They're not strong and vigorous. The leaves will be thin, the stems will be small, and that's a photomorphogenic response to poor light. If you have strong, high light, and the plant is a type plant that needs that, you're going to have a stronger plant, greater growth. We'll skip through a few things here as we move along, try to cover some stuff. As we talk about light, the usage of light by plants, the light receptor gene that we use is recognized as being what we call phytochrome, but that's the component that is responsible for the reception of light and use of it by plants. We also have a term called phototropism. And phototropism, again, if you break it down, photo means light, tropism means the movement or response of a given plant to light.
So a sunflower, something like a sunflower?
Well, we talked about sunflower just before you got in here, but yeah, that's sunflower is an example. But if you have a house plant in your house that's growing near a window, do you leave that plant alone and never change it? Just let it grow on its own?
No.
What do you do, Tyler?
Occasionally.
You move it occasionally. Why?
Because you don't want to be constantly bombarded by light being moved.
No, not exactly. You move it because most plants are going to grow towards the light. So you're going to have a plant that bends to get more light in that situation. So quite often you'll turn it around. So the plant will, again, move. It's a phototropic response. That's the movement of the plant in response to that light, a light source. And that's generally true we have with house plants. Another term that references light and the use of light, the term photoperiod. And photoperiod is a photomorphogenic response to variations in day length. We recognize that we have different categories of plants. Some are grouped as what we call long day plants. Some are short day plants. And then there's some called day neutral plants that really aren't affected much by it. But right now, if you go into a garden center or most big box stores, most any sort of establishment that carries plants, house plants, what's going to be the plant that you see predominantly right now?
Short day plants.
What is the one plant that's going to be on the shelf?
Poinsettias.
Excuse me?
Poinsettias?
A poinsettia, exactly.
Those are Christmas colored leaf plants.
Yeah, Tyler, that's what it is. Tyler said, was that the Christmas colored leaf plant? Yeah, but that's what it is. And poinsettia is a classic example of what we, excuse me, refer to as a short day plant. The growers of poinsettias have to manipulate the day length to have poinsettias available for the buying public during this season. If they were left alone to their normal and natural growth patterns, you wouldn't get the red bracts or the flower on a poinsettia until about mid to late January. So what we mean by, we recognize they're short day plants, and the places that grow poinsettias basically trick them. They will start giving them a short day around the 1st of October. And that's generally done by shade cloth or something or other to reduce the amount of light that they get during a given day. They want to cut it down to about 6.5 to 7 hours to make that plant begin to initiate flowers and begin to gain that color that's desirable for this season. But that's what we're talking about there, that 6 or 7 hours is referred to as the critical day length for whether we're talking about a short day plant or a long day plant. But a poinsettia is the one I always use as a point of discussion. And if you have a poinsettia at home that you have kept over from last year, if you've taken care of it, it's going to be a pretty plant about the middle of January, unless you do something or would have done something back in October. And that could be as simple as putting it in a closet or put it in a darkened room for 6 or 8 more hours than it would normally get.
That's what my uncle does.
What?
That's what my uncle does.
Okay. When does he start it? Do you know?
He started sticking. He has 3 of them. He started sticking them back in the closet shortly before Halloween, I think.
Okay. Well, that's about mid-October. Yeah, that's good. And the commercial greenhouses will have they have mechanized devices that pull darkened cloths over the rooms where they're growing these things. So. But that's what we're talking about with an example of short day. As we talk about plant growth and development, there's several other terms that you may or may not be aware of. The term dormancy, does that mean anything to anybody? When you talk about dormancy.
Dormancy is when something starts to cease a lot of its usual activities and goes into more or less a hibernative state.
You've got a good idea. Yeah. But dormancy is a temporary cessation of growth. And in the area in which we reside, the deciduous trees, in other words, the trees that drop their leaves, have currently gone into a state of dormancy, or they're approaching that right now. My grapevines have gone dormant. My apple trees have gone dormant. But that's a temporary cessation, in other words, a temporary stoppage of growth. And it's true in the region in which we reside with most of the broadleaf plants. Now, occasionally we've got a few broadleaf evergreens, but they don't do much growth. They still are basically dormant during this period of time. As we talk about dormancy, it brings up another idea that I'm going to skip ahead of myself. But anyway, trees like apples or peaches, once they go dormant, they begin to what we call accumulate growing degree days or chilling requirements, excuse me. They a given, well, let's use peaches as an example. Peaches are grown up and down the East Coast, all the way down into Florida. And if the varieties that are grown in Florida have a relatively low chilling requirement. In other words, they may only need 400 hours of temperatures below 40 degrees to meet their chilling requirement. Peaches that are successfully grown in our area probably going to need 12 to 1,300 hours. If you're looking at peaches that might be grown in Minnesota, they're probably looking at 1,800 hours. And this element of idea of chilling requirement is the amount of cold hours that are required by that plant to basically rest. And once that has accomplished that magical number for that particular variety, and you get temperatures or weather that's warm enough for it to begin to grow again, that plant will begin to grow and potentially flower. So if we were to take the varieties that are grown in Minnesota, move them to Florida, they would never flower.
No.
Because they can't get the number of chilling hours in Florida that it's required. And it's a mechanism that Mother Nature has built into these plants to enable them to survive. You can get a crop of peaches in Florida early because of this low chilling requirement, and those plants are adapted to that area. But if you move that particular variety to Minnesota,
Wednesday class.
by mid-December, early January, it would have met its chilling requirement. If you were to get a warm week, that plant would begin to flower, and all the production would be killed. So chilling requirement is a sort of a strange idea, but it's something that plant breeders use to make sure that plants are adapted to the area that they grow. It's necessary to break the dormancy that we were talking about. All right. Stratification is another term that relates to the accumulation of cold hours, and it's generally referred to as the period of time that's needed for a seed to meet this chilling requirement. I don't know what kind of noise we're getting, Tom. Sound like somebody scooting the chair.
I don't know. I heard
it's fine.
It sounded like a song.
Okay.
I don't know where it's going from. I blame
that. I don't. Doesn't matter. It's okay. But when we talk about seed germination, stratification is the requirement of a given seed to have an amount of a number of cold hours. An example of stratification, if we use a common weed as an example, a morning glory. If you have morning glories in your garden that grew this summer, and they come up with a little trumpet type flower, and the little seed pod develops, and it dries up, say the middle of July, early part of August, that seed will not germinate. It's a mechanism that Mother Nature has devised to prolong that species. So if the seed of the morning glory would germinate, say in September, it would begin to grow and be killed by early frost. But the fact that it has to be stratified or have a number of cold hours or chilling hours, that seed will have to lay over the winter and accumulate this number of cold hours before it will enable it to germinate next year. So, that's just one of the mechanisms that is done. Generally, we don't have that to be the case in most of our vegetables. Most of them will germinate and come up pretty quickly, even in the given season. But things like weeds and so forth, That's one of the mechanisms that's done with it. It's called stratification. The chilling requirement that is done by or needed for green and growing plants, sometimes we call that a vernalization, and that's a cold induction that will cause a flower to initiate. Sometimes people get excited with their gardening, and they may put in some rock and put it in a little bit too early, and in that situation, it may get chilled. And instead of producing a nice head, it will throw a flower spike. I've seen cabbage do that. Instead of producing a head of cabbage, it throws a flower spike. And that's a result of the vernalization, or the excessive chilling requirement, which causes it to, it's called a cold induction, producing that flower. All right, the next thing that we need to talk about as we talk about growth and development is water. And water is a key component in the growth of all plants. The majority of a plant, in most cases, there's going to be, depending upon the type of plant, but more than half, sometimes as much as 90% of a given plant is water. So that's why when we're talking about feed sources that you had in your animal science class, you would talk about the dry weight. That would be the weight of the material without the water. There's huge differences in water weight in given plants. But when water is plentiful, plants will grow more succulently. When it's limited, they tend to grow a lot more slowly. Cell walls will be a lot thicker, and they don't elongate. All right, as we talk about growth and development with water, two gases that are essential for growth and development that we talk about are the carbon dioxide and oxygen. And the opening and closing of stomate regulate the CO2 levels in plants, and it's increased greatly by photosynthetic activity. Oxygen occurs as a response to the respiration in plants. Respiration is the release of energy and the stored carbohydrates that are synthesized during the photosynthetic process. Carbohydrates are also referred to as sugars. When we begin to talk about germination, you've got an inhibition, which is the water uptake with a given seed. If you've ever taken some bean seeds, if you want to evaluate the potential for germination, you can put some bean seed, and I'm using that just because they're large and easy to visualize. But place those in between two pieces of paper towel and keep those towels moist. And you're going to start to grow. Well, you're going to see that seed begin to swell. That means it's taken up water. And as it continues to do that, the first thing that you see is going to be a little radicle, or the initial root, and it's going to begin to emerge. All right, I'm skipping over some stuff here. Begin to talk about shoots and roots. It says there are four principal functions of roots in higher plants. They are anchoring the plant to the soil, absorbing water and minerals, conducting water and dissolved minerals into the plant, as well as other organic materials. And then the roots are also storage mechanisms, such as sweet potatoes, sugar beets, and carrots. they store an awful lot of carbohydrates and sugars. Told you I was going to skip along pretty quickly. All right, we're going to talk about shoots. Shoot growth, in general annuals that we talk about, you're going to have two different types of shoot growth. And what I normally use for an example of this one is the tomato. There are distinct differences in tomato varieties. When we're talking about the differences in tomato varieties, you've got what we call determinate varieties, and then we have indeterminate varieties. And most of the heirloom or the older varieties of tomatoes that your parents grew and what I grow still are indeterminate tomatoes. And that is basically, the word defines this. It says it's indeterminate. It has not determined how tall my tomatoes are going to grow unless I physically prune them. Now, there are another family of tomato that are categorized as determinate tomatoes. It has been predetermined genetically how tall that tomato is going to grow. And this is a process that plant breeders have initiated to facilitate the commercial production of tomatoes. Most of the shipping type tomatoes that we get from now until probably the 4th of July that you have access to in the grocery stores are a product of what we call a determinate variety. Had a friend that, when I was at Tech, that worked under a plant breeder that was primarily working with apples and peaches, but Randolph, his name was Dr. Randolph Gardner, and he got a job at the Mountain Research Station down in Fletcher, North Carolina. And he was a tomato breeder. And Randolph was responsible for breeding what we call, the first variety that came out from there was Mountain Pride. This was a mountain research station, so he began to title his work. The varieties that he released were mountain varieties. And it was a determinate type tomato. In other words, it would grow up about 3 feet tall, maybe 40, 42 inches. And that's as tall as it will grow, no matter what you did to it. That's all the height you're going to get in that tomato. But it facilitated a lot of production in a small area. They would use in a string weave type culture in that, and they would plant successive crops. The commercial growers, they would, and they, because they didn't have to deal with excessive foliage. Now, the old heirloom tomatoes, things like German Johnson, Cherokee Purples, then things like Big Boy and Better Boy, they're all indeterminate tomatoes. If you plant those in your garden and you take care of them and stake them, they'll grow as high as a building if you in a good location and take care of those. But the height of those has not been determined. So that's two different categories of that we refer to as shoot growth to determine that. A lot of tomatoes are being grown in greenhouses now, and I have an opportunity to get an awful lot of hydroponic tomatoes to take to my market. Most of those right now are coming out of Canada, and they are a product of an indeterminate type of production. They're using more advanced varieties, but they want that plant, once they get it established, they want it to grow to the height of the ceiling in whatever growing structure that they're doing. Quite often they're harvesting those by the use of a stepladder, 6 and 7 feet tall. That's an example of an indeterminate as compared to a bush type or a determinate type. As we talk about shoot growth, we begin to talk about the different types of let me get to this. There's a part here I want to talk about. All right. There's another term here that we haven't talked about at all, and that's the use of growing degree days. Growing degree days is a way that we measure the heat accumulation for many plants. And growing degree days are needed for flower induction and the seed maturation in most of our annual plants. But a growing degree day, usually it's determined by a given base. And I won't spend a lot of time on this, but I think you need to have an idea of what it is. The maturity of given plants is based on so many growing degree days that a plant accumulates. And say we're using a base of 50, and the average temperature for a given day during the spring is 60 degrees. So if the base was 50, we accumulated 10 growing degree days if the average was 60. Had the average been 70, we would have accumulated 20 growing degree days. Now, assume we have a cool spell come in and the average temperature happens to be 50, we don't accumulate any growing degree days. So what we do is on a daily basis, you can evaluate the average temperature above a given base, and this is established for all sorts of crops, but it gets into a fairly technical method of determining when a given crop is going to mature. Done a lot for row crops that are done on a commercial basis. You know, if you had 1,000 acres of sweet corn and you knew that it took between 1,200 and 1,300 growing degree days to know when you needed to begin to harvest that, through computerization, you could, in weather instruments, you'd know that, come the 29th day of June, you could go start harvesting that crop without ever going to look at it. So that was one example. Skipping over a little bit of stuff here. Talks about pollination in this chapter, and it gives you a pretty good picture of a flower here where we're looking at figure 722, and it gives you the petal, the stigma, anther, filament, style, ovary, ovule. You need to be familiar with the way this picture is done. I just finished working on the test that we're going to have next week, and I will give you a hint. There is a flower on this the latter question in this test you're going to need to be able to identify some of these parts. So there've been some times when I didn't emphasize that as thoroughly as I am doing it right now, but make sure that you understand or familiar with the names of these structures that are in this flower diagram, figure 722. All right, another term that we talk about as far as fertilization is the term parthenocarpy, and that is the formation of a fruit without the stimulation of pollination or fertilization. It says without fertilization, no seeds are produced, but you can have a parthenocarpic fruit that are seedless. And examples of that are seedless cucumbers and seedless watermelons. They are parthenocarpic fruits. And they're induced in some plants by the applications of auxins and gibberellins. It says not all seedless fruits are parthenocarpic, but that's what we're talking about. We're going to talk a little bit about these hormones as we get a little farther. I don't think we'll get it done today, but we'll talk about it on Monday or Tuesday. One of the things it does mention here, it says the application of gibberellin to Thompson seedless grapes in their early development will increase the ultimate fruit size. Gibberellins is a growth hormone. It's a naturally occurring hormone, but there's been a lot of work done with the use of different hormones, both naturally occurring and some that have been synthetically done. When I worked in Winchester, there was a chemical company that was trying to work with some combinations of gibberellins and cytokinins to affect the shape of apples. This was back in the mid-'70s, early '80s. And it was Abbott Labs that were doing it. And the Washington State Apple Commission had basically brainwashed the buying public into the idea that a desirable Red Delicious had to be long, elongated, and have 5 prominent points on the calyx end. That's the picture that, if anybody said Red Delicious, that's what you're going to picture and visualize as to being the most desirable Red Delicious apple. Red Delicious that are grown in the East didn't have that type, elongated, typey shape that Washington State had built their industry on. And Abbott Labs had decided that there was a marketing opportunity for growth regulating hormones for the folks in the East to potentially put on their apple that would more exactly depict the Washington State type of apple. And they were working with a number of different formulations varying the percentages of gibberellins and cytokinins. And the material that they were eventually labeled was called promelin. But I was involved with some of the early work. We would have to treat the flower as it was blooming with different concentrations of these things. And it was really interesting as we worked along. They had some formulations that had a little bit too much of one compound or the other, and we got some Red Delicious that basically looked like red bananas. They would be small in diameter and 6 or 7 inches long because of the addition of these gibberellins and cytokinins. They had another formulation, another mix that we used that we sprayed on, and instead of it elongating the entire apple, it would elongate 2 or 3 of the points and not affect the others. And basically, those apples looked a little bit like a sheep or sheep's nose. It was a strange looking thing. They eventually came with the right formulation, and that particular compound has been used by progressive apple growers in the eastern part of the United States for a number of years. Naturally occurring compounds, it wasn't anything that was a detriment to anybody, but it was just a compound that was additional. There's been a lot of work with different types of wine grapes. Wine grapes tend to have tight, compact clusters, which in some situations is okay, but in other situations, if you happen to get a dry period and then a wet period, those tight clusters will take up a lot of water, and it'll actually rupture some grapes in the interior portion of that cluster, which will when you rupture the grape, you're going to get the collection of sugar and water and create a great opportunity for disease and begin to have rot occur in those grapes. So they've sprayed these gibberellins on certain wine grapes to elongate the bunches so they won't be as tight and as prone to disease. They haven't got that one nailed down real well yet. They're still playing with it. So, but these hormones that we're talking about are naturally occurring things that determine the amount of elongation that you have in cells as plants are growing. All right, another term, we mentioned it the other day, is one called senescence. That's a terminal, irreversible deteriorative change in living organisms, leading to the cellular and tissue breakdown and death. So if we're talking about senescence, if you had a rose in your lawn this summer, say it bloomed 1st of August, it was beautiful for 2 or 3 weeks. About the beginning of September, you notice that terminal portion began to deteriorate, to break down. That flower was actually senescing. In other words, it was deteriorating, and it changed the cellular makeup and make the type of that, so it would not be a beautiful flower anymore. But that's what we're talking about with senescence. And we're out of time again. All right, we'll try to wrap this chapter up on Monday and get you ready for the test 2, and then we'll have a final which will cover everything. Okay? If you got questions, send me an email.
Okay.
Thank you, sir.
Thank you.
Now, Tyler, what